Section 3 of Gray's Anatomy, Part 3. This is a LibriVox recording. All LibriVox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Recording by Julie van Mallegem. Anatomy of the Human Body, Part 3, by Henry Gray. Development of the Vascular System, Part 1. Blood vessels first make their appearance in several scattered vascular areas, which are developed simultaneously between the endoderm and the mesoderm of the yolk sac, i.e., outside the body of the embryo. Here a new type of cell, the angioblast or vasoformative cell, is differentiated from the mesoderm. These cells, as they divide, form small, dense syncytial masses, which soon join with similar masses by means of fine processes to form plexuses. These plexuses increase both by division and growth of its cells and by the addition of new angioblasts, which differentiate from the mesoderm. Within these solid plexuses, and also within the isolated masses of angioblasts, vacuoles appear through liquefaction of the central part of the syncytium into plasma. The lumen of the blood vessels, thus formed, is probably intracellular. The flattened cells at the periphery form the endothelium. The nucleated red blood corpuscles develop either from small masses of the original angioblast left attached to the inner wall of the lumen or directly from the flat endothelial cells. In either case, the syncytial mass thus formed projects from and is attached to the wall of the vessel. Such a mass is known as a blood island, and hemoglobin gradually accumulates within it. Later, the cells on the surface round up, giving the mass a mulberry-like appearance. Then the red blood cells break loose and are carried away in the plasma. Such free blood cells continue to divide. The term blood island was originally used for the syncytial masses of angioblasts found in the area vasculosa, but it is probably best to limit the term to the masses within the lumen from which the red blood cells arise, as Sabin has done. Blood islands have been seen in the area vasculosa, in the omphalocement vein and arteries, and in the dorsal aorta. The differentiation of angioblasts from the mesoderm occurs not only in the area vasculosa, but within the embryo, and probably most of the larger blood vessels are developed in situ in this manner. This process of the differentiation of angioblasts from the mesoderm probably ceases in different regions of the embryo at different periods, and after its cessation new vessels are formed by sprouts from vessel already laid down in the form of capillary plexuses. The first rudiment of the heart appears as a pair of tubular vessels which are developed in the splanchnople of the pericardial area. These are named the primitive aortae, and a direct continuity is soon established between them and the vessels of the yolk sac. Each receives anteriorly a vein, the vitelline vein, from the yolk sac, and is prolonged backward on the lateral aspect of the notochord under the name of the dorsal aorta. The dorsal aortae give branches to the yolk sac, and are continued backward through the body stalk, as the umbilical arteries to the villi of the chorion. Ethernet describes the circulation in an embryo, which he estimates to be about 13 days old. The rudiment of the heart is situated immediately below the foregut and consists of a short stem. It gives off two vessels, the primitive aortae, which run backward, one on either side of the notochord, and then pass into the body stalk, along which they are carried to the chorion. From the chorionic villi, the blood is returned by a pair of umbilical veins, which unite in the body stalk to form a single vessel and subsequently encircle the mouth of the yolk sac and open into the heart. At the junction of the yolk sac and body stalk, each vein is joined by a branch from the vascular plexus of the yolk sac. From his observations, it seems that in the human embryo the chorionic circulation is established before that on the yolk sac. By the forward growth and flexure of the head, the pericardial area and the anterior portions of the primitive aortae are folded backward on the ventral aspect of the foregut, and the original relation of the somatopleur and splanchnopleur layers of the pericardial area is reversed. Each primitive aorta now consists of a ventral and a dorsal part connected anteriorly by an arch. These three parts are named respectively the anterior ventral aorta, the dorsal aorta, and the first cephalic arch. The vitelline veins, which enter the embryo through the anterior wall of the umbilical orifice, are now continuous with the posterior ends of the anterior ventral aorta. With the formation of the tail fold, the posterior parts of the primitive aortae are carried forward in a ventral direction to form the posterior ventral aortae and primary caudal arches. In the pericardial region, the two primitive aortae grow together and fuse to form a single tubular heart, the posterior end of which receives the two vitelline veins, while from its interior end the two anterior ventral aortae emerge. The first cephalic arches pass through the mandibular arches, and behind them 5 additional pairs subsequently develop, so that altogether 6 pairs of aortic arches are formed. The 5th arches are very transitory vessels connecting the ventral aortae with the dorsal ends of the 6 arches. By the rhythmical contraction of the tubular heart, the blood is forced through the aortae and blood vessels of the vascular area, from which it is returned to the heart by the vitelline veins. This constitutes the vitelline circulation, and by means of it nutriment is absorbed from the yolk vitalis. The vitelline veins at first open separately into the posterior end of the tubular heart, but after a time their terminal portions fuse to form a single vessel. The vitelline veins ultimately drain the blood from the digestive tube and are modified to form the portal vein. This is caused by the growth of the liver, which interrupts their direct continuity with the heart, and the blood returned by them circulates through the liver before reaching the heart. With the atrophy of the yolk sac, the vitelline circulation diminishes and ultimately ceases, while an increasing amount of blood is carried through the umbilical arteries to the villi of the chorion. Subsequently, as the non-placental chorionic villi atrophy, their vessels disappear, and then the umbilical arteries convey the whole of their contents to the placenta, whence it is returned to the heart by the umbilical veins. In this manner the placental circulation is established, and by means of it nutritive materials are absorbed from, and waste products given up to the maternal blood. The umbilical veins, like the vitelline, undergo interruption in the developing liver, and the blood returned by them passes through this organ before reaching the heart. Ultimately, the right umbilical vein shrivels up and disappears. During the occurrence of these changes, great alterations take place in the primitive heart and blood vessels. Further development of the heart. Between the endothelial lining and the outer wall of the heart, there exists for a time an intricate trabecular network of mesodermal tissue from which, at a later stage, the musculi papillaris, chordae tenonii, and trabeculae are developed. The simple tubular heart, already described, becomes elongated and bends on itself so as to form an S-shaped loop, the anterior part bending to the right and the posterior part to the left. The intermediate portion arches transversely from left to right, and then turns sharply forward into the anterior part of the loop. Slight contractions make their appearance in the tube, and divide it from behind forward into 5 parts, viz. 1. The sinus venosus, 2. the primitive atrium. 3. The